guys, this is Mikey from Avo Prep Academy. Today we're going to do something a little bit different. Instead of having a more of a lecture video, I decided that we should maybe take a look at just an FRQ section from, I don't know, I just drew 2019. These are all publicly available on the College Board website, so I presume that there's no copyright issues or anything like that with me showing you guys this on the video. But the idea is, yes, I know you guys haven't gone through the entirety of the course yet, so some of this content you may have not have covered. But it is true that majority of AP Biology students had taken ninth grade biology, where you probably have covered things like molecular biology, genetics, and ecology to a certain degree. So I don't think the content that we'll cover here will be lost. And I do recognize that some schools actually go through AP Biology in a slightly different order than the one that's offered by the course exam and description PDF. So the units could be shuffled. So I thought maybe, since many of you guys have been asking about more videos on solving these questions, that I would tackle this 2019 FRQ. And I'm not going to actually write on this because I'm actually just at my office right now, so the setup is slightly different. I'm using an OBS for the first time, so I don't exactly know how this is going to work out. But the answers will be left in the description as a PDF, so you guys can look through that and see what I would have written for each of these answers. But I'll try my best without writing on the actual PDF to describe what is happening. So hopefully this will be kind of a useful thing for you. Oh, and by the way, a bonus is that a lot of school teachers actually use these old exam questions as unit test questions too. I'm not sure if that's because they just expect students never to look these up, but I've seen that happen before. So, and some of my friends do that. So if that's the case, then, well, hopefully this will help you out. So let's take a look at 2019, and we're going to look at question number 1 first. So this one is about a metabolic pathway. And the first tip here is that anytime you look at an AP Biology exam, like, you're going to read this stuff and it's going to not make a lot of sense. It's, obviously it's not a reading test, so they're not trying to be as obscure as possible, but, you are looking at words you've never seen before. So one of my advice is to actually just look at the figure first and try to see what's going on here, what units that you guys are being asked to decipher. And the first thing that I noticed that this is a metabolic pathway. And a metabolic pathway is something that we're introduced to in Campbell chapter 8 on energy and enzymes. And that's what we see with tryptophan being converted into I3PA, being converted into IAA. Whatever those things are, it doesn't really matter. We have this metabolic pathway, a very short one. But layered on top of this is molecular biology, because in, I think, chapter 17, which is called Genes to Proteins, we learn about transcription, which is the action of DNA being transcribed onto messenger RNA. And then we have translation, which is the RNA being manifested into a protein through ribosomes, and that will be translation. So what AP Biology likes to do is to layer multiple chapters together, because at the end of the day, what's happening inside a cell is a cell, right? The cell doesn't care about whether something that they're doing is in Unit 1 or Unit 4 of AP Biology, obviously. So we have to take everything as synthesized into a singular, I guess, cohesive story. Okay, so let's take a look at what they're saying then. Auxins are hormones. They're plant hormones that coordinate several aspects of root growth and development. They tell us that IAA is an auxin, so that'll be the final product here, that is synthesized from tryptophan, which makes sense. And then the next sentence is really about what enzymes are involved in the production of I3PA from tryptophan, and then, of course, IAA from I3PA. That's it. That's all we have to know. So let's take a look at the first question. And by the way, a lot of these questions are relatively easy. We do have one or two tricky components of these, A, B, C, D, E subsections. But the first one says, circle one arrow that represents transcription on the template pathway. We've already sort of talked about this, but when you have a gene that is then copied onto a messenger RNA template, that will be transcription. And because they tell us to circle one arrow, I'm going to circle either this arrow or this arrow, but either one is okay. Now, some students always ask me, do they take off marks if you circle both arrows? I don't know, because to be honest, we get emails all the time asking us to come down to the grading center and to volunteer. Actually, it's not volunteers paid, but to grade AP exams. I've never actually gone and done that myself, but I do know from experience that each grader does grade separately. Follow the instruction. I would say that that's the best way to go. If they say circle one arrow, just circle it and you're good to go. And then they say identify the molecule that would be absent if enzyme YUC is non-functional, well, if enzyme YUC is non-functional, then this arrow goes out the window, and what would be absent would be IAA, because I3PA would still be there. In fact, you would actually have more I3PA because, well, it's not being converted into IAA. So all you'd have to do is just write a sentence here that says, if enzyme YUC is non-functional, then IAA would be absent. Do you have to explain it? Not so much for identify, but I always like to do it just because I want to tell my students, in the PDFs of my answer keys, like this is why it's happening. But for the purpose of the exam, you just have to identify that. Okay, B says predict how the deletion of one base pair in the fourth codon of the coding region of TRPT would most likely affect the production of IAA and justify your prediction. So in this scenario, we're actually looking at mutations, which is still part of that chapter 17. And in that chapter, what we learn is that there are many types of mutations. There are mutations that change a base pair, or there are mutations that either insert or delete base pairs. And one of the things that we always like to emphasize is that deletion of base pairs, or a one base pair, or many base pairs for that matter, usually results in something called a frame shift mutation. And frame shift mutations are rather dangerous because if you remember the concepts of how DNA and its subsequent RNA transcripts are translated into amino acids, each of the amino acid in a sequence is represented by 3 bases. So we take these 3 and say, look, this is the reading frame. We have AUG, and then we have CGC, and we have GGC, and so on. But if you delete one of them, especially in the fourth codon, out of hundreds of codons that presumably would make up an enzyme, then you're basically shifting the entire reading frame fairly early on, which means that all of the amino acids that would follow, including the fourth codon, by the way, all the amino acids that would follow, including the fourth codon, would most likely be incorrect. If they're correct, it would just be by accident, right? And because the amino acid sequence is so important in terms of how the structure of that protein folds, you're more likely than not to get a misfolded protein, which then would result in a non-functional protein. And I would say that the justification part should include that idea of how structure and function, or form and function, are correlated, because again, in this case, because we have the erroneous genetic code, we're going to misfold the protein, and because the protein has a wrong structure, it's going to have a wrong function, rather a non-function, indicating that it's not going to perform its duty. And of course, if TRPT is broken, then, or if the enzyme TRPT is broken, then we're going to have a reduction in I3PA. And if I3PA as a reactant to the next step is unavailable, then that's going to severely affect IAA production. And that is the idea behind it. Now, like I said, the PDF that I've linked below is going to have a more formal sentence like answer for this, and hopefully you guys can look at that and see how you would formulate this question, the answers to this question as well. Let's take a look at the next one. Explain one feedback mechanism by which a cell could prevent the production of too much IAA without limiting I3PA production. So in this question, we are going right back to that chapter 8 where we talk about feedback mechanisms. Negative feedback mechanisms are common in biology, obviously, and in this case, that's the one we were looking at. It's a negative feedback because you want to prevent overproduction. That's always the case in negative feedback. You have something that's being produced too much. Typically, that substance inhibits either an enzyme or some pathway that would lead to the production of that substrate. So in this case, specifically, they're talking about IAA. So how do we prevent too much IAA production without limiting I3PA production? So we are given a bit of a caveat, as in, look, that IAA should not affect anything prior to I3PA, meaning that we're most likely, the most obvious answer is that IAA is involved in a negative feedback mechanism that could inhibit enzyme YUC. So to explain that simply, if you have too much IAA, it maybe binds to an allosteric site on the enzyme YUC that alters the active site of that enzyme, thereby making it inactive. And as a result, I3PA is no longer produced into IAA, which then self-regulates the amount of IAA that is present in the cell. Now, while that is probably the most obvious answer from chapter 8, I'll tell you that chapter 18 actually has something to say about this as well. Now, if you haven't gone through chapter 18 yet, chapter 18 in Campbell Biology, the big form textbook, that's regulation of gene expression. And the reason that that's important is that another possibility is that IAA could potentially downregulate the expression of this gene from the actual DNA to RNA. So maybe it, let's say binds to a substance that limits the activity of the transcription factor that binds to the promoter of gene YUC, or just any sort of imagination, any sort of thing that you could imagine that would feed back into the entire pathway, and that includes, let's say post-transcription regulation. So, for example, pre-translational regulation, another way to think about it too, is that IAA could inhibit the translation of that messenger RNA even if it's produced. So there are many, many, many different points at which we can regulate the expression of this enzyme. But ultimately, either the presence of the enzyme or the activity of that enzyme, it needs to be regulated by IAA in order for us to answer this question properly. Okay, let's look at the next one, which sort of takes us away from molecular biology, it looks like, and probably into ecology. Let's take a look. Rhizobacteria are a group of bacteria that live in nodules on plant roots. Rhizobacteria can produce IAA and convert atmospheric nitrogen into forms that can be used by plants. Okay, so they actually have a couple of things that they do. They can stimulate the root growth, which is interesting. Okay, and they also are nitrogen-fixing bacteria, which are interesting because remember that in symbiosis, specifically mutualism that we learned in ecology, one of the more used example is legumes, like soybeans, for example, and they have something called root nodules, and they host nitrogen-fixing bacteria. And the reason for this, by the way, if we wanted to go back to a little bit of background information, is that even though nitrogen is really abundant as a gas in our atmosphere, plants can't really use any of the gaseous nitrogen. They have to use nitrites or nitrates that are in the soil after that gas is converted into usable nitrogen through a process called nitrogen fixation and so on. But because nitrogen can be limiting in many, many terrestrial ecosystems, over time, evolution has resulted in certain plants being able to form this really intricate symbiosis by hosting nitrogen-fixing bacteria themselves on the part of the plant that we call the root nodules, which can assist in these plants alleviating that nitrogen limitation. So let's take a look at what else they say here. Plants release carbon-containing molecules into nodules. Okay, so that would mean that in return for the job well done by the bacteria, plants can give the bacteria sugars, and of course carbon-containing molecules, mostly sugars, so that they can feed the bacteria. So it's a good win-win situation. Identify the most likely ecological relationship between plants and rhizobacteria. So that would be mutualism. One of the things that I would be careful of is just using the word symbiosis. Symbiosis includes mutualism, but it also includes things like parasitism. So you probably don't want to conflate the terms symbiosis and mutualism. So mutualism is a type of symbiosis, and in this case, it's very specific. Both parties win. Describe one advantage to bacteria of producing IAA. So as this text shows, it says IAA coordinates root growth and development. So clearly, if the bacteria are able to produce this auxin and stimulate root growth and development in their homes, it's kind of like home improvement, right? They're able to stimulate plants to produce more prolific roots, which would allow them to actually gain nutrients, other sources, not just nitrogen, but things like phosphorus and potassium. Better for greater photosynthetic capacity. That's always good for the recipients of those sugars, and it also probably allows them to proliferate to a greater population density because they have more root nodules on that plant. So all good answers there. The last one says, a researcher removed the plant nodule and identified several cheater rhizobacteria that do not produce IAA or fix nitrogen. All right, so this is a common problem in evolutionary biology, isn't it? So they have these cheaters that are reaping the benefits of all of the actual nitrogen fixing bacteria, but they don't do anything. It doesn't cost them anything. But frequency actually matters, though, because take a look at what happens here. It says, describe the evolutionary advantage of being a bacterial cheater. So I think I've already done that, because if you're in a population of predominantly non-cheater bacteria, you can sort of skate by, right? You can be you can be cheating, and because everyone else is not cheating, you just sort of reap the benefits. But the problem here, as I said, is that it has to be predominantly non-cheater, so you can sort of be in the shadows. Because here's what happens, though, later if plants recognize something is wrong. If there are too many cheaters, let's say, because it says plants can adjust the amount of carbon-containing molecules released into nodules in response to the amount of nitrogen fixed in the module. This means that the plants are not dumb, right? They're not going to just sit there and be like, well, all of y'all are cheating. I'm just going to give you guys sugars without testing anything. No, they are going to ensure that they're getting their fair share, and that's going to regulate how much carbon is produced. And they're not doing this actively, right? Plants don't have brains, but all through the course of natural selection, let's say plants who were, let's say, naive and just gave them all the sugars, regardless of whether the bacteria were cheating or not, would have been weeded out. So some sort of molecular pathway would have evolved, and it allows plants to recognize predominant cheating populations and react accordingly. Now, it's a predicted change in bacterial population that would cause the plants to reduce the amount of carbon-containing molecules. Now, this actually is a really silly question because they literally told you that if the amount, actually they don't tell you, so I guess you have to assume. So if the amount of nitrogen that's in the nodule decreases, then presumably that's going to reduce the amount of carbon-containing molecules. I mean, I sort of explained that already in my previous statement, assuming that it is pretty obvious that the amount of nitrogen that's being changed is going to be for the worse, right? And that is going to reduce the carbon-containing molecule. All right, so that would be the evolutionary pathway that would preserve the plant's ability to detect cheaters, not preserve, but allow that type of mechanisms to regulate the amount of loss. Okay, so number one's not bad, not bad. So it's a little bit of ecology, a little bit of molecular biology, and a little bit of, metabolic pathways. All right, let's take a look at the next one. And here we're looking at protists, and it looks like populations, right? Because take a look at this. This graph shows, not graph, but this table shows that you have time over here on the left, and then you have group 1 where species A and B are grown separately, group 2 grown together, and we have the population numbers, 1,500 for species A, 700 for species B grown separately and then grown together, 1,400 for species A, 0 for species B. Ooh, okay, so what's going on here? So let me give you guys a little bit of background on this question. So first and foremost, we are looking at, I think they tell us this was paramecium, right? No, they don't tell us it's paramecium. But the reason that I say paramecium is that one of the most common examples that we use for teaching the concept of population growth affected by competition is in paramecium. So I think there's paramecium caudatum and paramecium aurelia. I think I'm pronouncing those species properly. If not, then just comment below and let me know. And essentially the idea is that if two organisms, or not two organisms, but two populations of species that have substantially overlapping niches coexist in a system, then one of them is going to go extinct. And generally speaking, if we just say generally, or if we're using words a little bit more generally, I would say that the more competitive species is going to win, and the less competitive species is going to go locally extinct. Now, Tillman's theory of competition is the conceptual framework that would model this type of relationship. That's something that you guys can look up later. But the idea is, again, for AP Biology, we don't need to involve a lot of math into this. Just know that if there are two very similar species and their populations coexist in a same area, the more competitive one, whatever that might mean, let's say more resourceful or more resilient, the more competitive one is going to, let's say, drive the other species to local extinction. And that's precisely what we see in these two columns where they're grown together. Species A clearly is more competitive. But the other concept that I want to bring to your attention is that even though species A wins eventually, you can see that competition is just bad for even species A. I mean, clearly it's bad for species B. But we see in, let's say, ecological relationships that competition is a minus-minus relationship, as in it hurts both parties. It's not like capitalism where, you have competition and the consumer wins. Like there's no winner, or there are no winners in competition in ecology here, at least in the general context. So obviously you can see that 1,400 is lower than 1,500 at 70 hour because competition sucks for species A too. Okay, so let's take a look at some of these questions. The growth curve of species B for group 1, so that one, and group two species A, so that one have been plotted. Plot the other two and label the graph. So I'm not going to necessarily do that in this video, but basically the non-shaded one you want to lay on top of the graph that they've drawn here. Clearly, if we want to use the labels properly, you should probably look at something like 1600 being the maxima over onto the y-axis to accommodate for the largest number that we see, maybe around here on that 1400, and this goes all the way to the tip of the graph in that case. So that's all we would have to do for the graph in question. The x-axis will be time, so hopefully you guys are well familiar with that. All right, let's take a look at some of these concept questions though. B says, as shown in the table, the student established treatment group 2 with 5 individuals of each species. Provide reasoning for the reduced initial population sizes. So in this case, they're asking why this group here only has 5, whereas this one has 10. It's just a control to ensure that the population density in the vessel, whatever vessel that they're using, is the same, whether you're looking at them separately or together. I know that there are probably better ways to control for that, but in AP Biology, that's all they're asking for. It's a control to ensure that the population density is the same in both conditions, group 1 and group 2. And then the student claims that A and B compete for the same food source. Provide 2 pieces of evidence from the data that supports the student's claim. So in this case, we are going to have to refer back to the competition idea from ecology that I mentioned. So I would argue that you can say, well, when you look at these organisms separately, so let's say when you look at species A and B grown separately, they do better, but when they're grown together, one of them goes locally extinct. This would mean that they have overlapping niches and they're sharing the same resources. And essentially, that would be the basic claim. But the answer key, if you actually take a look at the answer key or the grading rubric, they go into the very specific details of, well, the growth rate is faster in group 1 than group 2. Okay? Grows to a higher population in 1 than in 2. It's like these are all the same ideas. So I get a little bit irked when College Board does this, where they ask you to, specifically make 2 claims, but it really can be boiled down to one major claim, and then you would just break it off into, I would say, smaller streams of that idea. So again, the PDF that I upload is going to have some ideas as how you approach that. All right, predict 2 factors that will most likely limit population growth of species A in treatment group 1. So this is just talking about a group that's grown separately. So one of the things that we notice is that the population follows what we call the logistics growth curve. So from ecology, logistics growth curve is one in which you have a slow, gradual increase in the population until you get to an exponential phase. And then you have something called the transitional phase where the growth slows down, and then it reaches something called carrying capacity, or the Kmax. And that's what we see here. Now, typically, we have 2 different types of factors that we can mention, one of which is density-independent factors and density-dependent factors. Because this is a question talking more about protists in a closed vessel, I would probably focus on density-dependent factors, maybe talk about, limitations on food. So that's one. Limitations of space. That's another one. Increased pathogen infectiousness within more dense populations is another. There are a myriad of examples when it comes to density-dependent factors that regulate population growth. So any of those would be okay here. And then the last one, this is fun because as soon as I see contractile vacuole, I'm like, okay, this is a throwback to Unit 2 where we talked about membranes and also things like, osmosis. Because in osmosis, we saw that water, given that there's like an aquaporin or some passage through which it can traverse, it's always going to move to an area with lower osmotic pressure, which is generally associated with, let's say, greater solute concentration. Many protists are freshwater, as in they live in an environment where it's clearly going to be hypotonic. The environment has less solute. So you have a lot of water that constantly comes into the organism. So they developed a vacuole that swells up and pumps that water out constantly. It's like our heart, pumping all the time. For them, it just pumps all the time. The student repeated the experiment using a growth medium with lower solute concentration. So we're actually making this even more hypotonic than it is. So how does the activity of the contractile vacuole change in this new condition? Well, it would actually increase because remember now the differential, that's the word that I'm trying to say here, the differential between the solute concentration of the environment and the cell would be even greater, meaning that even more water will rush in and the rate, or at least, will increase, and you would need more contractile vacuole action in order to pump that water out. Okay, all right. So numbers 1 and 2 are generally the big questions, and they seem pretty okay from 2019. So lucky students from 2019. Let's take a look at the next one. We can go through the rest a little bit more quickly. It says the pyruvate dehydrogenase complex, or PDC, catalyzes the conversion of pyruvate into acetyl-CoA, a substrate for the Krebs cycle. The rate of pyruvate conversion is reduced in individuals with PDC deficiency, a rare disorder. Well, that all makes sense. Where is this from? This is from Unit 3, Energetics. Specifically, I think it's Chapter 9, where we're going to cell respiration. The pyruvate is a molecule that is produced at the end of glycolysis, which, as you guys know, happens in the cytoplasm. Pyruvate is imported into the mitochondria via a process called pyruvate oxidation. And during that process, a carbon dioxide falls off of a pyruvate molecule, coenzyme A takes its place, and you get acetyl-CoA. That happens in the mitochondrial matrix. So that's where that is. And by the way, I can link a video or pages from the textbook that can tell you exactly what drawing that they're referring to. Make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle. So pyruvate converting into acetyl-CoA, that's what PDC does. That's happening after glycolysis. So would this disease of PDC deficiency affect any of the processes that are upstream in the sequence of events? And the answer is no. So NADH produced by glycolysis most likely not affected. Now this, of course, is not taking into consideration like feedback loops, and it's like, oh, there's not more of this, so it increases glycolysis and all that. No, just looking at the most simplified model, NADH should not be affected, or production should not be affected in glycolysis. But the amount of NADH produced by Krebs cycle certainly will, because remember that acetyl-CoA goes, in the simplest sense, acetyl-CoA feeds into the Krebs cycle, or at least the 2 carbons anyway feeds into the Krebs cycle. So if we have less acetyl-CoA, we're going to have less Krebs cycle, we're going to have less NADH. That ultimately reduces the amount of ATP being produced, and that would be why this is a problem, right? It's a problem for people with this disorder. And the reasoning is, as I mentioned, the positioning of this reaction. It's downstream of glycolysis, but upstream of Krebs cycle, and that would be one of the best ways to think about it. Now, C says PDC deficiency is caused by PDH1A mutation, which is located on the X chromosome. Ooh, now we're looking at genetics. A male with PDC deficiency and a homozygous female with no family history of PDC have a male offspring. Calculate the probability that the male offspring will have a PDC deficiency. I hate doing genetics by talking, but here's one thing that I will say. This woman, or the mom, the female here, will have 2 X chromosomes with no PDC deficiency, so she'll be completely normal, and that's the chromosome that she'll give to her son. The male with the PDC deficiency has that deficiency on his X chromosome, but a male offspring never gets that X chromosome from the dad. A male is a male because it has a Y chromosome, at least biologically speaking, right? So the dad is giving the Y chromosome to the son here, and that's not going to have that PDC gene at all, so the probability is likely 0, right? And this is 0 because there's no family history of PDC deficiency. If they tell us that the mom has a father who had PDC deficiency, then she for sure is a carrier, as long as she herself does not have that disorder. She's a carrier, and then the rate here would be like 50%, right? But no, that's not the question. So we're going to go with 0%, and that'll be the cleanest answer for number 3. Cool. Let's go to number 4. 2019. This is after 2014 when they got rid of physiology from AP Bio. So this is actually a little bit tricky, right? Some teachers will actually go through the nervous system a little bit, especially when you talk about membranes and stuff, because sodium-potassium pump is so intricately tied to action potentials and so on. But if your teacher has gone through some of these ideas, that's awesome. If your teacher had not gone through these ideas, you're going to have to make the best inference from what they tell you. So let's take a look. All right, so we have a neurotransmitter. That's acetylcholine, by the way. That's what they're telling you. Acetylcholine is a neurotransmitter that is released from the presynaptic neuron into the synapse. It diffuses through the synapse and binds to these ligand-gated ion channels, or the neurotransmitter receptors, on the postsynaptic neuron. Of course, pre and post meaning before and after the synapse. That totally makes sense. Once this neurotransmitter binds, it opens these channels, which then allows usually sodium to flood in, making the inside of the cell very positive. I mean, think about it. Sodium is a cation, is positively charged. If a lot of that comes into the cell's membrane potential will increase. And then you have this decrease from potassium ions going out. But the idea is that this action potential will relay a signal. Now it says a researcher is investigating the effect of a particular neurotoxin that causes the amount of acetylcholine released from the presynaptic neuron to increase. Okay, so that's actually an interesting neurotoxin. Instead of diminishing some effect, it's actually increasing the amount of acetylcholine. But that's not always a good thing, and let's find out if they ask about that. Describe the immediate effect of the neurotoxin on the number of action potential in a postsynaptic neuron. So if the action potential is caused by acetylcholine binding to the receptors, then it should increase, because you would have more acetylcholine that is likely to stick around and continuously bind to the ligand receptor. So by the way, if there's any relationship to the things that you may have learned at all, this would be chapter 11 on cell communication, where we talk about the importance of concentration. Like, the concentration of these ligands are going to then allow the continuity of ligand binding to activate these receptors. Okay. And it says, predict whether maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. Now, I don't think there's any way to answer this without knowing a little bit of neurobiology. So I'm a little bit irked by this question. The answer is that it's going to stay the same. And the rationale for that is, as I mentioned, if you look up here where the maximum membrane potential is reached, this is when these potassium channels open, allowing the flooding out of potassium that decreases your membrane potential. But again, I don't know how you would know that, unfortunately. Let's take a look at B. The researcher proposes two models, A and B, for using acetylcholinesterase, an enzyme that degrades acetylcholine to prevent effects of the neurotoxin. Okay, so that makes sense. If that breaks down acetylcholine, increasing that would probably work against that toxin. In model A, acetylcholinesterase is added. In model B, it's added to the cytoplasm. Why the heck would you do that? Right? We can clearly see from the diagram that acetylcholine is binding to the receptors, and they're not lipid-soluble messengers that go in. So the one in the cytoplasm would have no effect. You would have to put it into the synapse, and that would be the rationale for this answer. Okay, moving on. Number 5. Evolutionary biology. So here we have the divergence table of mitochondrial DNA sequences. All right, so by the way, just so that you guys remember, mitochondria are, they arose through endosymbiosis, and they have their own bacterial-like genome. We can actually sequence it, and we can look for differences between mitochondrial genetic data between similar species, and we should expect to see differences that could resolve relationships, even among similar species. That's because mitochondrial DNA tend to mutate a little bit faster than some of the heavily selected regions of our genome. And that's what we see. But remember that in this case, we're looking at divergence, so differences, right? So the smaller these numbers, the closer we are. And we know this already, that humans and chimpanzees are the closest siblings, followed by, let's say, humans and gorillas, and then humans and orangutans, and humans and gibbons. Like, we can kind of see that. And we can also see this at a zoo. You look at chimpanzees, they look a lot closer to us. And then as we go through gorilla, orangutan, gibbons, they sort of look farther and farther. And I know the morphological information or anecdotes are not always going to be the most accurate, but in this case, it is. And hopefully, you guys have some experience in these primates. So based on fossil data, the researcher estimates that human and their most closely related species in the dataset diverged approximately 7 years ago. Using these data, calculate the rate of mtDNA percent divergence per millions of years between humans and their most closely related species in the data set. All right, so the humans and the most closely related species, this is, of course, in reference to chimpanzees, and it says 7 million years ago, and they want us to calculate percent divergence per million years, So we just have to divide this 8.8 by 7, and that should give us how much divergence there would have been per million years. I think that's about like 1.2 something, 1.25. I think that would be a decent enough answer. Using the data, construct a cladogram on the table. So I'm just going to do this right off the top of my head. That's humans, that's chimps. By the way, you can put chimps and humans here. I mean, that's kind of weird because we are humans and whatever. But humans, chimps, or chimps or humans. And then gorillas on the third line and orangutans on the fourth line. And then when they say, why are the gibbons as the outgroup? Well, it's sort of a tautology because literally the outgroup is the most, I guess the least related species to the in-groups over here, right? So the gibbon clearly in this case has the most amount of divergence from any of the other organisms in this case. So we would put, or species, so we put the gibbons as the outgroup, and you can see that we would have humans, chimps, and gorillas and orangutans on the inside. So it's a tautology because we put it there because it's literally the most different, and then it's the most different because we're able to put it out here in the cladogram. Draw a circle around all the species that are descended from the node within the square. That would, of course, be, in this case, humans, chimps, and gorillas. And that ancestor would be the ancestor of all of us, chimps, humans, and gorillas. And that's one of the reasons that we say that these species are not our ancestors, but rather our cousins, because we just simply happen to share great-great-great-great-great-great-great-grandmother or something to that degree. Okay, moving on. This is actually a really tough question. I remember this. All right, so here I think we're looking at Saccharomyces cerevisiae, which are yeast that can live as haploids, but can also converge together. Not converge, but they come together as a diploid for sexual recombination. In this case, we're looking at how certain mutant strains of Saccharomyces can either produce amino acids or unable, or they might not be able to produce amino acids. So let me actually just walk you guys through this. In treatment 1, in the medium, we have all of the amino acids that an organism would ever need. The wild type obviously can survive. The mutant 1 and 2 can both survive because all of the things they need are there. But if there are no amino acids, you can see that the regular wild type Saccharomyces could actually synthesize all of the amino acids and make do, whereas mutant 1 and mutant 2 cannot. So this actually tells you that we don't know which amino acid or acids each of these mutants cannot produce, but we know that it's at least one of them, because you take away everything and they're unable to survive. Now we're going to get a little bit closer to what that might be. It says if you put all amino acids except methionine, well, I mean, logically wild type should be able to survive. If they can make any amino acids from nothing, clearly they can make methionine even if there is none. Mutant 2 seems like they can make methionine, but mutant 1 cannot. Right? So even though you give mutants all the other amino acids, all 19 other amino acids, if there's no methionine, they can't do it. They can't survive. They can't make methionine. But on the other hand, they can make leucine, because mutant 1, you can see that if you present all amino acids except for leucine, they can happily make leucine all day long and be fine, but mutant 2 cannot make leucine. Okay, so now we know that mutant 1 is deficient in their ability to make methionine. Mutant 2 is deficient in their ability to make leucine. Role of treatment 1 is, of course, in this case, the control. We want to ensure in this positive control that all of these mutant cells can actually survive given all amino acids, so that the reason that they're dying here isn't because of some other reason other than the amino acid issue, right? Because if they didn't survive here, then clearly it's not the amino acid that's inhibiting their survival, it's something else. But we can verify that, hey, look, if you give them all amino acids, they do survive. So this must have something to do with the absence of methionine. Provide reasoning to explain how mutant 1 can grow in treatment 1, but cannot grow in treatment 3. Well, in treatment 1, in this case, they have all amino acids, but in treatment 3, they're missing methionine. And given what we've talked about, with the mutation most likely inhibiting methionine formation in this mutant 1, that would be the best answer. And again, PDF in the link below. Yeast mate by fusing two haploid cells to make diploid cells. In a second experiment, the researcher mates mutant 1 and 2 haploid strains to produce diploids. Using the table provided, predict whether the diploid cells will grow on each of the four media, using plus signs to indicate growth and minus sign to indicate no growth. All right, so now this is where things get fun, because I feel like we can overlay some of these ideas with genetics as well. So for instance, all amino acids present, the diploid version of mutant 1 and 2, when you combine them together, that's of course a plus. Treatment 2, that's actually also a plus. And I'll tell you why. Because mutant 1 can't make methionine, but they can make leucine. Mutant 2 cannot make leucine, but they can make methionine. When they're together, they can make methionine and leucine by relying on each other. So while mutant 2 allows for the production of methionine in their genome, mutant 1 allows for the production of leucine. So even though each of them are missing one, together they can help each other out and actually produce what else is, or what each of these guys are missing. So that's a plus, and that's a plus, and that's a plus. Because as we said, diploid means that they can offset each other's faults. So that is a really cool idea that it's actually, I think it's actually a very famous science experiment. I can't remember what it is right at this point. If I remember it, I'll put it on the description. But it's a very famous experiment looking at Saccharomyces with these enzymes. Okay, next one. So what we're looking at here is a very interesting figure. On the column here, we have all the different tissues: liver, heart, brain, kidney, pancreas, skeletal muscle, and then we have these different genes. And these colors, or not colors, but the degree to which these blocks are shaded indicate the amount of RNA presence in the cytoplasm. So the most obvious takeaway is that the more RNA that you have, the more mRNA that you have, the more protein expression there is. It doesn't always have to be that way, but that's the idea. And it says, based on the data provided, identify the gene that is most likely to encode for a protein that is an essential component of glycolysis. Here we go again, right? Glycolysis. They love this. They love the idea that glycolysis is universal, not only across all organisms, but also within an organism. Because remember, glycolysis is that first step in producing energy. And part of what defines a living organism is that they have the ability to metabolize, which means to convert things into energy. And if we're talking about glycolysis, it has to happen in every single one of these cells. So gene G right here that is present in every single cell, or at least not the gene, but the RNA is present in every single cell, that is the best candidate for glycolysis. Because why the heck would liver, let's say that glycolysis, one of the genes, gene H was involved in that. Like, why would it be the case that liver and heart does not perform glycolysis? That doesn't make any sense. It has to be every single one of those tissues, and that's the reasoning. Now it says, you could have high levels of gene H mRNA, let's say something like the kidney, but not always have the protein. So how would that happen? So this is something that I mentioned earlier, that you could get something like pre-translational regulation. So going back to this drawing, you could have a lot of the mRNA, but if there's something that's regulating the translation of that RNA into the proteins, that potentially could downregulate the actual protein concentration, while the mRNA would still be detectable in the cytoplasm. So this is chapter 18, and they mentioned a couple of these possibilities. One of them is something like small interfering RNA and microRNA, and these are RNA molecules that have the ability to bind to messenger RNA and either mark it for destruction or inhibit its interaction with the ribosome. So you have this ability to downregulate protein translation even though the RNA is present. And this is, I guess, if the teacher teaches that in chapter 18, which would be fantastic, that's fine. The students will have no issue. But I do find that many curricula don't include that part. So it's almost an inference question for many, many students. So unfortunately, that is the case. But hey, you've heard it here. All right, so let's keep moving on. The last one seems like it's about, membrane transport. So this is unit 2, going all the way back. I'm not going to go into the background too much because I do want, this video is getting pretty long, but the main idea is that pH is acidity or alkalinity. It's associated with H plus ion concentration. So you can see that in the vacuole, there's more H plus here. There's fewer H plus, so the pH goes up. It becomes more alkaline. That makes sense. Flower color, I guess at low pH is red and higher pH is blue. Cell volume, small, large, that probably has a little bit more to do with water going into the plant cell. But let's take a look. The petal color of Mexican morning glory changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin. Ooh, I like that. Anthocyanin is found in the vacuole of petal cells. So that's the vacuole. The petal color is determined by the pH. Okay, the model of morning glory petal cell before and after flower opening is shown. So, by the way, if you're actually taking this test on the day of the test, you probably don't have a lot of time left. Let's just tackle the question. Identify the cell component in the model that is responsible for the increase in the pH of the vacuole during flower opening. Boom. So that's going to be a proton pump, right? Because protons move H plus ions. And in this case, increase in the pH. Oh, I'm sorry, that's not true. Ooh, I almost made a mistake. That's potassium proton transport, because the increase in the pH would be the movement of these protons out of the vacuole, and that seems to be happening with the K plus H plus transport protein. So that's my bad. Describe the component's role in changing the pH of the vacuole. So again, it's trading potassium for protons, and as protons come out of the vacuole, you actually have higher pH because it becomes less acidic and more alkaline. A researcher claims that the activation of K plus H plus transport protein causes the vacuole to swell with water. Okay, provide reasoning. All right, so that seems reasonable. Earlier we just had H plus ions, now they're leaving. That's okay, but there's a lot of potassium coming in. Potassium is just like anything, it's a solute. So as the solute concentration of the vacuole increases, it becomes more. It becomes greater in osmolarity. So it becomes more concentrated, and water simply follows. And this is literally what we see with the movement of water in these arrows. And boom, there you have it. That's number 8. Like I said, I'm going to post the PDF of all the of my answers to this at the bottom of the description so you guys can take a look at it. But it's not bad, right? Like, there are definitely units that you guys probably studied that's not going to come up on the FRQ, and you're going to be pretty mad. You're going to be like, hey, I spend so much time looking at crossing over or gene mapping, and like none of that comes up. Circadian rhythm, none of that comes up. It's worse now because there are only 6 questions, but it is true that there are themes that repeat time and time again. And one of the things I noticed is that in this 2019, there's actually a little bit less emphasis on evolutionary biology, except for that phylogeny question. But it depends, right? It depends. And the most important thing is that you just have to know everything, I guess. I know that's not encouraging. But to do that, keep watching our videos. So, hey guys, if this was useful, then, comment. Let me know. Let me know if you want more of this, because I can do, 2014 all the way to 2023, and we can do these videos and hopefully help you guys out. But if you haven't done so already, subscribe, like the video, and I guess turn that bell icon on, all that YouTube stuff. This is a little bit more informal video. Like, I'm not standing in front of my Sony camera, so I feel a little bit more connected, so to say. So that's kind of fun too. But yeah, we'll see you next time. Bye.